Hello, good afternoon. Welcome to biology class. In the previous classes, we have discussed about some aspects about plant breeding. That is about how to develop the plants against some of the diseases, maybe caused by bacteria, maybe caused by viruses, maybe caused by funguses like smut, rust, etc. In today's class, we shall discuss about the plant breeding for developing resistant to the insect pest. Okay, the plant must become resistant for the insect pest. What is the word pest? When you use the word pest, it means that it is nothing but one of the harmful organism. Pest means harmful organism. The pest may be an insect, pest may be another plant, pest may be a bacteria, pest may be one of the viruses, any organism which is harmful to our crop plants. So, I am growing a jowar plant. In our field, I am growing jowar. Around jowar plant, whichever the plant coming and affecting the jowar plant, it is called pest. Okay, either one bacteria is affecting the jowar, bacteria is the pest, or virus is affecting the jowar, virus is the pest, or some insects are attacking on the jowar plant, then insects are also called the pests, right. So, here we shall discuss how we can develop the plants against the insect pests, because in nature there are various insects. So, insects are either useful or harmful. Useful insects like our honeybees, our butterflies, etc. So, they are useful for the plants for the process of pollination. They are useful for the process of pollination. Instead of useful, there are many more harmful insects like take mosquitoes, aphids, jassids, stem borer, ball worms. So, all these insects are harmful for the crop plants. Okay. Not only this, many more are there. Still more insects are there. Those are all harmful for our crop plants. Okay. Whatever the crop you are growing, the crop will be affected by these many insect pests. So, our aim of the plant breeding is to develop the plants against the insect pest, the plants resistant to the insect pests, right. So, we all know the common methodology of the plant breeding. So, here first we need a source organism. Source organism means source of resistance. Means for developing the resistance we need some source. The source may be either cultivated plants or the source may be the wild varieties. Means the resistance genes may be taken from other varieties of cultivated plants, or they can be taken from the wild varieties or wild relatives of these cultivated plants. Okay. Then what actually is the resistance? Right. We said resistance. What does the resistance mean here? Resistance of a host. Here, resistance in the plant, in the crop plant means nothing but modification in morphology, physiology and other aspects, or biochemistry, morphology means you have heard about trichomes, hairs on the stem, right? All those are the modifications. Physiology, change in the respiration, photosynthesis pattern, etc. Biochemistry, production of some chemicals, like that. All these, if they are modified, then the plant will be resistant to those insect pests. The plants will be resistant for those insect pests only if they are modified in these aspects. Okay. So, we shall see the methods how these are developed. Firstly, we can see by the morphological modifications naturally, these are naturally modified. So, there are some modifications in the plants, and those modifications, because of those modifications, the plants are resistant to these insects. Okay. Firstly, we have hairy leaves in cotton, means on the surface of the leaf in cotton there are small hair like structures, what we call trichomes here. Okay, trichomes. So, those hair like structures are helpful for the cotton plant to be resistant for these insects, seeds and or leaf hoppers. Meanwhile, the hairy leaves in wheat, because of these hairs on the wheat plant is resistant to the cereal leaf beetle. Similarly, solid stem in wheat. Remember, the stem may be hollow or it may be solid. Hollow means inside the stem there is a cavity. Solid, inside the stem there is no cavity. Inside the stem there is no cavity. So, hollow stem, solid stem. Okay. So, if the stem is solid, then the plants will be resistant for stem saw fly. Okay. Saw means one of the cutting instrument used by the carpenters. Similarly, the insect has the mouth parts like a saw. So, if the stem is solid, then the plant will be resistant for the stem saw fly. Then in cotton, if leaf is very smooth, oily, then nectarless, this nature will protect the cotton plant from bollworms. Okay, we will discuss again this detail about this in biotechnology. Next chapter we shall discuss again how this affects the cotton, how this can be controlled by biotechnological techniques. We shall see in the next concept of biotechnology. And high aspartic acid content, high nitrogen content and high sugar content, low nitrogen and low sugar in maize will protect from maize stem borer, means the insect which attack on the maize plant. Like that, these modifications, morphological, physiological modifications or the biochemical modifications in these plants will protect them from the respective insects. Okay. I think in your book they have just given a paragraph. So, I wish you can all just make a list of this in the form of a table, and it will be easier for you to remember for the examination. You may have one of the 1 or 2 mark questions on this. Okay. Name the modifications in a wheat plant which are helpful for the wheat plant for resistant to these insects. Then you can write the modifications. Right. Now, we shall see how the resistance can be developed by plant breeding techniques. Okay. What are the methods used to develop resistance against many insects? So, methods or steps. Here, the resistance can be developed by following steps. Firstly, the screening of germplasm. Screening of germplasm for resistance sources. Screening of germplasm for resistant sources means we should identify which plant has the resistant genes. Okay, among that you can once choose which one is resistant, which one is not resistant. After choosing, you should subject it for hybridization. Hybridization among the selected plants. Means you should select some plants which are having resistance. Among those selected plants, you should undergo that cross pollination, emasculation, bagging, whatever you discussed in earlier classes. Then after hybridization, then you should go for a testing for insect pest resistance. If it is tested good for the resistance, then you can release of new varieties. Because before releasing any crop variety, you must test for the resistance. Whatever we discussed here, resistant to many more insect, we have to test for it. Okay, whatever you have done earlier, that must be tested. Okay, before you bring that into the market, a plant must be tested for the resistance. Then only you can publish it as a new variety. Then only you can publish it as a new variety. Okay, so here we have some of the crop varieties which are resistant to many more insects. Okay, so you can see the Brassica, it is also called Indian rapeseed. So, or rapeseed mustard. So, variety is Pusa gauro. So, Pusa gauro variety of Brassica is resistant to the insect group called aphids. Okay, remember this variety of this crop is resistant to this insect. Okay, you should read like this. Don't read like this. Pusa gauro variety of Brassica is resistant to the aphids. Similarly, Pusa some 2 and some 3 of flat bean are resistant to the seeds, aphids and fruit borers. And lastly, Pusa 7th and Pusa A4 is resistant to the shoot and fruit borer. These are varieties of the okra, what we call lady's finger. Okay, so these varieties of these crops are resistant to these insect pests. Okay, so by following these methods, the varieties of various croplands are developed so that they can get resistant to those insect pests. Right, that's how we can study the development of the croplands resistant to the insect pests. Okay, here the important part is to remember the examples. Okay, methods are already common. Okay, just wherever you go, just add this word. Other than that, everything same. Okay, wherever we get resistance, other things are same. Okay, in this concept, this you have to remember. You will get one or 2 mark questions out of this. Okay, you cannot expect 5 mark question in this concept. You can expect a 2 mark question in this concept. Right. We shall now see the development of the crops with improved food quality. Plants or crops with improved food quality. Plant breeding for improved food quality. Plant breeding for improved food quality. Here, whenever we say the word food quality, food quality means the diet or the food must contain adequate amount of carbohydrates, adequate amount of proteins, and healthy fats, many more micronutrients, many more minerals, essential oils, and mainly the vitamins. So when all these are present in optimum quantity, use the word optimum or minimal amount of these compounds in our food, then the food will be healthy or balanced. The food will be balanced, right? But nowadays, due to increasing population, the food quality is decreasing. Food quality is decreasing day by day. Some people are not even getting sufficient amount of the food. Okay, many people in the slums area, in the small villages, or many more places, many people are not getting sufficient food. Okay, that's what is the hidden hunger nowadays. Okay, hidden hunger is the major problem in our country. In some places, not in all the places. In some places, there is a hidden hunger just because of the food quality and the imbalanced food or less improved diet. Okay, so here if the diet is deficient in many more in these molecules, it may lead to many problems. Okay, here the mental ability decreases, right? And the overall growth of the child or any person decreases, or it may lead to many diseases. Means chances of attacking of the pathogens will be increased. Like that, many more problems may arise if the diet is deficient in these molecules. Okay, so only there is a branch of biology that deals with improvement of the food quality. That is what we call by name biofortification. Okay, it is nothing but breeding of crops crop plants for improving the quality of the food in terms of protein, oil, vitamin, etc. Simply plant breeding for improving food quality. Okay, you can say in simple terms plant breeding for improving the food quality. Okay, quality means all these biomolecules, all these essential molecules. Okay, so what are the objectives? Objectives of biofortification. The objectives include improvement of the following 4 molecules, mainly protein content and quality. Means in the food the content and the quality of the protein must be more. Secondly, oil content and quality. Oil content and quality. Thirdly, vitamin content. And lastly, the 4th main objective of the biofortification is to improve micromolecules and minerals. Macromolecules and minerals. So these are the 4 main objects of plant breeding on mainly the biofortification. Okay, when the food has all these, food follows all these 4 aspects, then our diet will be healthy. Okay, in the IARI New Delhi, in the IARI means Indian Agricultural Research Institute. Many more varieties of many crops are developed. We shall see what are those. So in 2000, year of 2000, maize hybrids are developed. Maize hybrids are developed. And those maize hybrids contain essential amino acids like aspartic acid, and tryptophan. Because the cereals like jowar, wheat, bajra, rice, they are poor in these amino acids. Okay, so our aim is to improve the quality of these amino acids or the amount of these amino acids in the food. So these maize hybrids contain these molecules or these amino acids in a more content. And one variety of wheat, Atlas 66, this contains a high amount of the protein. High amount of the protein. One more, iron fortified rice. Iron fortified rice. This contains 5 times more iron than normal rice varieties. normal rice varieties. Okay, like that there are many more food molecules with respective vitamin or the biomolecular content. Okay, and students, you have a small homework. You should come actually here at IRI New Delhi. IRI New Delhi. There are some 4 categories of croplands developed for improving some biomolecules. I will just write down the molecular content. You should have to write the croplands. Vitamin A enriched croplands you should mention. Vitamin C enriched. Vitamin C enriched. Some croplands are there. Iron and calcium enriched. Some crops are there in our text. And lastly, protein enriched. Some crops are there. Just open this concept in our textbook. In the last page, the last few lines, last 4 lines have this concept. Okay, in the IRI New Delhi, they have developed some plants, some crop plants which are enriched with these molecules. Okay, just note down those things and add in today's assignment. Add in today's assignment. Krishna sir will give some assignment questions. Okay, okay, he may give 4 or 5 questions. Along with those 4 or 5 questions, you have to add these 4 into your daily assignment. Okay, so today assignment will contain questions given by Krishna sir and also the answers, means the crop plants rich in these molecules. Okay, just open the biofortification page and go to the last 4 lines, you will get the answer for this. Okay, in next class we shall see about the single celled proteins. Single celled proteins. Till that, keep learning. One more thing, you are not sending the assignments on time regularly. Please do follow the assignments regularly. It's not just a punishment for you, it is an improvement for you. Okay, please make use of that facility given to you, writing and sending the assignments daily. Means you are attending the classes regularly. Okay, we shall see in the next class. Till that, keep learning. Take care. Bye bye.